Traditional DNA databases depend on one critical factor to work. The suspect has to already be in the system. If they aren't, the physical evidence hits a dead end. To solve cases that have sat dormant for decades, forensic science had to stop asking, "Does this DNA match someone?" and start asking, "Who are you related to?" For decades, crime scene samples simply sat inside evidence lockers, unusable if they were too small, heat damaged, or degraded by time. But a process called next-generation sequencing, or NGS, changes how we treat older samples. Instead of relying on pristine samples, scientists use NGS to extract usable genetic markers from microscopic, heavily damaged traces of biology, like a single skin cell or a trace of saliva on aged clothing. Investigators assemble those markers to build a digital genetic map and upload it directly to consumer ancestry databases. They aren't trying to find the suspect's direct profile
Of that same genetic code online. The database rarely returns a direct hit. Instead, it identifies distant relatives, like second or third cousins scattered across multiple states, who share small portions of the suspect's biology. This initiates a process of building up. Forensic genealogists use these distant cousin matches to construct a family tree backward through time, tracing the individual branches across several generations. They follow these lines upward until they find the historical convergence point, a set of common ancestors, like great-grandparents. Once that historical convergence point is found, the investigation shifts into reverse branching. Investigators map out every single descendant flowing forward in time from those common ancestors. This naturally generates thousands of names. To find the suspect inside that massive web, they begin the elimination phase.
As investigators evaluate historical public records, like the detailed demographic data visible on this 1930 federal census schedule, to establish strict timelines. They cross-reference those names with death certificates, marriage licenses, and historical newspaper obituaries to track where each person lived and died. As investigators apply parameters like birth year, known geographic location, and deceased status, entire bloodlines are ruled out. The net slowly tightens, systematically eliminating branches until a clear pattern emerges. Applying this reverse branching logic, we reach a final, isolated household of 4 siblings. We know the original crime occurred in Chicago in 1995, and the DNA profile indicates a male suspect. Look at the parameters and determine the target. Node A was born 5 years after the event. Node B does not match the biological profile. Node D lived overseas. By process of elimination, Brother C is isolated as the primary target. Through public records, the biological trace left at the crime scene is no longer anonymous. It is explicitly tied to a verified identity, a specific family, and a confirmed geographic reality. Genealogy points investigators in the right direction, but they verify their target before dedicating resources. To do this, they rely on a process called SNP analysis, or single nucleotide polymorphisms. Because DNA is predictive, scientists translate the genetic code into a highly probable physical profile. It generates a digital estimation of facial structure, eye color, and skin pigmentation, giving investigators visual confirmation that the person they mapped matches the biology. As this graphic illustrates, genealogy cannot convict a suspect in court. The legal system strictly requires a direct, legal one-to-one DNA match between the suspect and the original crime scene evidence. Investigators use surreptitious sampling to covertly collect discarded biology, like a coffee cup, which is legally abandoned property. Sequencing this newly collected DNA and comparing it to the decades-old evidence locker sample confirms the one-to-one match, completing the investigation. This is how forensic genetic genealogy operates. Investigators extract dormant evidence with next-generation sequencing, build a family tree from distant kinship matches, reverse branch downward using public records, visually confirm the target with phenotyping, and close the case with a final surreptitious sample.